Okay, so
Yes.
we are live.
Yeah, hello everyone. Nice to have you both here, Dimiter and Sai. It's a pleasure to be at my channel and have this conversation. This is not a debate, it's just to have a friendly chat about origins, abiogenesis, the origin of life, to exchange some ideas and information. And then I hope that this can be a progressive exchange of ideas and information. So I will remove myself from the screen and I will leave you both for having this conversation. And thanks to everyone which has appeared at my channel to follow what you guys are saying. I've not asked for how much that this would be, but I would say for about an hour and a half, depending on how it goes. And I leave that as well to you both to decide for how much you want to keep that conversation. When you end, then I will join again and either invite people from the chat that can come in and continue, or eventually close and finish the chat, depending what seems to fit better. Okay, so I leave it to you both and have fun.
Should we introduce ourselves first? I guess that would probably.
Yeah, that's okay. So go ahead, say a few words.
Oh, okay. Well, so I am a PhD biochemist. I'm retired from working, although I still like to do some theoretical work when I can. My experience actually was in environmental toxicology, molecular biology, carcinogenesis research, nothing to do with our current topic. But I've had a long interest in the origin of life, just sort of an amateur interest. And I am especially interested in a particular question, which is how does the fidelity of replication impact on the origin of life? And I'll go into much more detail on that. I recently had a paper accepted. It's a theoretical paper. It's still in press, but I did send a copy to Dimiter. And I'm still working on that, but I'm pretty much a amateur compared to him. So he has sent me 3 wonderful papers that he has published, and I read them with great interest, and I will talk about this more later because I found them really very much in tune with my own thoughts about the origin of life, which is an amazing subject for science. It's really, I don't know. Now that I'm old and retired, I wish I had been able to go into it as a actual, experimental researcher much earlier. But trying to catch up. I'm also a Christian, and I'm interested in the nexus between science and Christian faith, but I'm not sure that will come into this discussion. I don't see how it, unless there are questions. I don't know. If there are questions, then we'll answer them. But
there might be later questions. Yeah,
it could be for a different topic. But
I'm a little bit envy on you because first of all, you have enough free time to play and enjoy.
Yeah, that is true.
I don't have that luxury here.
I'll tell you, you have something to look forward to because being retired is great. You can actually do exactly what you want, which, as you know, is not the case when you're an active scientist.
You have to get crossing my fingers.
Yeah, you have to get. People don't know this. You have to get grants. You have to serve on committees. There's so much. And anyway, we won't get into that now. But
also, the second thing that I'm envy, you're kind of good at mathematics. I'm not that good at mathematics, unfortunately. Just the basic stuff, but that's what it is. So
I'm not that good. I'm still a biologist, and none of us are very good at math.
But anyway, so. Other than this, I'm a molecular biologist, and all my career was dedicated to cancer research.
Oh, okay. Like me.
And especially DNA replication
Okay.
is my field, the cell cycle, DNA replication, cell cycle, DNA repair,
Right.
and all of that intersections which are related to cancer. And of course, that's nothing to do with origin of life, but as a researcher, that's what it was. In respect to origin of life, it was kind of very strange how I came to these papers, actually, because initially, from long time, even from my student years, I, of course, this is interesting area, and I always was very interested in origin of life, but I never thought that I'll be involved in any way. And also, to me, the researchers was doing their job, and obviously, in all of my career, I didn't even thought that I might come up with something that it's not published yet. Because back in my mind, the people working hard for that, and presumably they have a kind of grants to do that. They are paid to do that, so they're serious people. Probably approximately, maybe by now it's 6 years ago or something like that. Such a period of time, I debated with different creationists, and everybody knows that in such a debate, always you come to the origin of life.
Right.
So, and what happened that the the story. I mean, I'm not going to go in details right now, but long story short, I always told them that there is a way to information to appear just by stability of RNA. So, and especially when you have 3D structure of RNA, could be more stable or not. And that was debate, and after all, I simply stopped debating with creationists at all. At that moment, because first of all, I was quite busy and that was not even important at all. However, meanwhile, I realized that I simply cannot stop thinking about. Even that I'm no longer debating, even I'm not in any relation and anything, I simply was not able to stop thinking about. And because of my background of molecular biology, I decided just to think about and try to make my own ideas and concepts about that. And it was not too much of effort to me, but it kind of took me approximately 2 years or something like that, just nothing special. But constantly just coming back to me, and so on. And finally, I come and crystallize some idea, some concept for origin of life. And I said to myself, oh, that's not too serious, or at least I know that it's serious, but what I didn't know that. So what is the actual, at the moment, published in the topic? Because during the time when I was reading, when I was just trying to pick up and make my own hypothesis for origin of life, I didn't read too much about, because my molecular biology background, how the RNA behaves, how peptides behave, all of that was just in my background. I didn't need to read something special for this. And suddenly, and after all, when I have some concept idea, I decided, okay, let's see what the people actually have published. What is in the field? After all, there is a scientists that are working on this field. And they have a grants, they have a institution, someone paid them to do that. So they should be good at that. So my expectation was just to see my own hypothesis in some way, by formulate it in one way, another way, with different words, somehow to be formulated someone from somewhere. So I started to read. Actually, now I started to read the field to see what's there. I read lots of papers, probably around 150 papers. Everybody was saying something. Everybody was discussing something. And suddenly I realized that, wow, my idea is not there. So what to do? I kind of trapped myself to my own whatever is there. So from one perspective, I can say, okay, I have something in mind, and that something in mind obviously solve some problems in the origin of life. On another side, there is a publications which to me doesn't make too much sense and does not solve so well the problematic. And I decided, okay, so what the heck? So let's go and publish. However, it's like a key that you have opened the door. You open the door, but immediately after that, there is another door and another door, and you just keep opening the door with the right key. And that's why it's accumulated material, lots of material it came. And everything came after another. And that's how I came with the publishing of those 3 papers. However, the last paper that I published was in my mind the very first. So, but I keep it to publish it at last. So
that's the one on the general topic that says point of no return.
Yes.
Yeah.
So
yeah, I really like that paper. That was a really good summary, I thought.
So the actual definition for life, the definition and what is important for origin of life, was the very first thing that I tried and I was working with 3 years ago. So, but it happened that's how it happened to be the last paper to publish. So if someone want to read the paper, especially my papers, I would recommend to start with the very last one first. After that, to go to the very first one, and after that, to go to the second one. It's simply, that's how it logically follows. That your paper is wonderful as well. I mean, I was very happy to see this paper. It makes kind of very interesting concept because it's mathematical model.
Thank you. Yeah.
And it says very important stuff there. I would say it's a very simplified model.
Very simple.
And also in each parameters, for example, if you take just the stability, for example, probability for stability, this PS, for example, you can have many interactions, many things there, which either in a linear or non-linear fashion might influence this PS.
Exactly, right.
And that's why it's just I accept this paper as a kind of very good summary of the concept of the idea. So if you don't mind, you can present briefly your paper, what is there, and what kind of conclusions you have from your paper.
Okay.
It is just suggestion. from myself, and after that I can present my stuff, and also I can try to discuss with you where my stuff and hypothesis or concepts can enter in your paper, basically, as a, so how your paper can respond to my hypothesis and to the RNA world hypothesis as well.
Yeah, before I talk about what my paper says, I just wanted to, I when I was reading your first paper, the first one to read, even though it came out last, I actually copied some of your phrases, which I completely agree with, and I think are really wonderful. I just want to mention a few of those. One of the things you say is the establishment of heredity defines the emergence of adaptation and diversification toward more complex structures, i.e. Darwinian evolution. And that's, I think, the central theme of everything I'm doing is that we don't pay enough attention to heredity. When people say, what is evolution? You hear, well, it's natural selection. Yes, that's true. It's mutation and variation. Yes, that's true. And sometimes they say heredity, and sometimes they don't even bother. They just forget it.
Yes.
And in fact, the mechanism of heredity is the key to evolution, in my view. And if you think about it, and I think you'll agree, there's nothing else in the universe that self-replicates other than living cells. A living cell, and that's amazing. I mean, that's an enormous thing, that you have this living cell that makes copies of itself, and it doesn't just make copies of its DNA; it makes copies of the entire cell. So, the image I like to use is, suppose you see a house that you really love. You go into it, you love it inside. It's a great house. You want to have an exact replica. So you tell an architect, build me the same house. I want the same plumbing, the same electricity, the same heating system. And I also want all the same books, all the same pictures on the wall, all the same furniture, all the same clothing, all of that.
Yeah.
Okay. That's what a cell does. And it does it by itself. If you wanted to make a copy of the house, you could do it. You need an architect. You need to start from scratch. You need to build a house. The cell makes itself. There's no house that can replicate itself. There's nothing that can replicate itself. So you have to think about how the cell does this. And it has to do it if you're going to have evolution. Because just think of it this way. Let's say you have an animal, a cell, an organism, doesn't matter what kind of organism, and it undergoes a mutation, and that mutation gives a better phenotype. In other words, it improves the cell in some way, or the organism. Maybe a bird has better eyesight, or a bacterium has a better
beneficial. Right.
That's correct. So that's selectively good. But when the cell or the organism divides, or if it's an organism when it gives birth to new organisms, if that change is not accurately transmitted to the new generation, there's no evolution. Okay, so if a bird has a mutation and now has better eyesight, that bird's going to do great, right? It's going to eat a lot, it's going to reproduce. But if the kids don't have it, it's over. That bird eventually dies and nothing happens. So
that's why I just for one sentence, and you'll continue. That's why I used to say that for evolution, it doesn't matter are you survived or not. That's not relevant. Exactly. What is relevant is who and what will give the offsprings.
Exactly.
The offsprings are important, and who will give his genes next generation. Okay, continue. Sorry that I disrupted.
No, I'm glad you said that, because sometimes when I read evolutionary papers or evolutionary comments, it drives me crazy, because people say, well, this happened and that gave them an advantage, and so it spread through. How does it spread unless they reproduce, right?
Exactly.
So I discussed this in a way, my story is similar to yours. I've been talking to, I was also in cancer research. I mean, that's where the grants were, right? So I never did this professionally, but then I had this interest, and I remember talking to a colleague, a friend, who is one of the, big people in origin of life research. And I raised the issue, and I said, how could you get evolution started unless you already had, a pretty good inheritance mechanism? He said, well, you could have had evolution with an error, with a lot of errors, a sort of an error-prone kind of replication. And you mentioned that a little bit in either your second or third paper, where you talk about early translation mechanisms, which are really fascinating. But I don't think we'll be able to go into it right now. So I said, well, that's very interesting. That's certainly true. I mean, I don't see why you couldn't have, for example, peptides and small proteins that are pretty good but not great. And if there's some changes, okay, you could still get things working. But then I started wondering, how much accuracy do you need? How good does it have to be? Now, today, in all life of all kinds, the accuracy of replication is 99.99999%.
It's very good. Yeah,
extremely good, both for replication of DNA and for translation to the phenotype. Very high accuracy. So, okay, obviously life didn't start that way, right? So the question that I asked was, how low can you go in accuracy, in fidelity? In other words, how high can the error be before you run into either no accurate replication of a mutation that causes a selective advantage, or before you hit an error catastrophe? And error catastrophe is, as you know, is when there are so many errors in any of these processes that the cell dies because it's not making the right proteins.
And that's actually proven experimentally very
well.
well.
That's a well-known phenomenon, right? And it's a strategy, for example, for antiviral therapies. You force the virus to mutate to the point where it's no longer functional. So that was my question, and I decided to ask, okay, let's say you start with one cell, it divides. Let's say it has a fitness of some kind, which I ended up calling probability of survival. And as you just said, that includes everything. That includes, all the molecular biology that has to go on to allow a cell to live, energy production, metabolism, everything. And today, our modern cells, that value is close to 1. I mean, cells don't. It depends on the environment, of course, but generally cells live, have a fairly high probability of survival before they divide, depending on the environment. But anyway, but then I asked, okay, now let's see what the error rate is. And the question is, given their probability of survival, their overall fitness, and various levels of accuracy in replication, what happens? Yeah. The first cell divides, gives 2 cells, 2 cells give 4, 4 give 8, et cetera, et cetera. So everything is fine. You end up with 2 to the n, where n is the number of generations of cells. And that's what usually happens when bacteria are growing in a dish, et cetera. However, what I put into this was, okay, let's say your accuracy of replication is not 100%. Let's say it's 80% or 60% or 40%. What does that do to the cell count number? And that's what the model was that I developed and that I used, and I got some very surprising answers. Just to summarize it quickly, it turns out that you could actually, and I didn't expect this, if the cell is extremely fit to begin with, you could have no accuracy of replication at all, and you will still get some survival. Cells will grow very slowly, but they're not going to wipe out. But if you go down in probability of survival, in other words, if the cell is not almost 100% fit, you've got to increase your replication fidelity a great deal.
Yeah.
And I ended up with getting all the data. This is from simulations. I used the Monte Carlo method with, and the details are in the paper. But basically what I found was that there's no continuity. And now this brings up another subject in evolutionary biology. You know, Darwin originally said that every step has to be gradual and continuous.
Yes. Yeah.
And that's been disproven in a number of cases with things like endosymbiogenosis. There have been some.
Yeah, it's not necessary to be exactly that. So
Exactly.
you can have jumps.
Yes, saltations, exactly. And there are many examples of those. And what I found was that that's also the case for this issue. In other words, if you start with a very low degree of fitness and a very low degree of replication fidelity, you can't improve. Okay, there's a level where it's not gonna, you're never gonna get evolution. The cell line will actually die out if it ever begins. As you go up, okay, there's a point at which it's almost like a phase transition. There's a point at which now you can evolve. So if you start with, say, 70% of fidelity and 70% fitness, no problem. The cell can evolve to what we have today. But if you start at 40% of each, and there are very different levels of the two parameters, no. So it's not a continuous starting, let's say, with 10% and then slowly goes up, up, up. It doesn't, that can't work. There's a discontinuity there. And that, I think, is important because it applies to it. I'm not doing the kind of work you're doing where you're actually looking at the mechanisms, okay, for translation, for the genetic code, for, you know, how information evolves. I'm saying it does. This applies to anything. This applies to any of the mechanisms you find. There's a point at which you have to have a certain level of replication accuracy, and below that level, it doesn't matter. You're not gonna be able to get evolution going. And getting evolution going, that's the key thing, because before you get evolution, you could have cells metabolizing like crazy. Great, wonderful. Eventually they go away, they die. You know, that just by second law. I mean, the thing about evolution is it's able to overcome all, as Dawkins said, right? It's that what does he call it? The crane. Okay, so evolution, because of the power of natural selection, can overcome all kinds of obstacles. But when you don't have evolution, when you don't have accurate replication, and you know, mutation and all the other mechanisms, you're not going to get life.
Yeah. I mean, also this is so two things just very quickly. A friend of mine, Mike, which is from our circle of discussion circle, he wants to know a little bit more details about your program that you've been using, and he wants to, because he is kind of programmer, and he can also go in simulation. So I shouldn't forget. So eventually, when we finished our discussion, I wish we to continue to have our kind of conversations and links. So
I can send you the program. It's a MATLAB program.
So if you don't mind, I can send you, for example, some link, whatever we have, just to continue our work.
Sure.
Also, the second thing, I see that as a populational mathematics or modeling, populational genetics. This is not new as idea, and I even saw some of the papers. Unfortunately, I cannot citate right now, so that's not important. But the people doing such a similar modeling and especially even for the ecological purposes.
Right.
For example, by such a calculations, you can figure it out a specific population of animals or species. At some point, when this population became too low as a numbers,
Right.
they are in the verge of extinction.
That's right.
And that's why this population genetics and modeling is very popular among the ecologists.
Yeah.
And
Yeah. That's
they
right.
know very well that a certain species, when the number drops to some level, they need to be kind of harbored or carry on or taking care of those just to expand artificially to expand the number in order to save the species.
That's right.
And that's the people know very well from very similar to your models. As I said, I don't know the details right now because I don't know details of many things and I cannot be everywhere. So that's two things that I just want to mention. So probably you already saw similar models as yours. Probably you did your research well.
I have a lot of references in the paper. Not as many as you have. You have a real
Yeah, I
very
spent lots
extensive
of time.
reference.
I spent lots of time to read and to find. I was so as a scientist, I have a principle of mine. If you have hypothesis, your first goal is to destroy your hypothesis.
Right.
That's the very first thing you have to do. Destroy completely your hypothesis. Find a way. Be devil advocate from everywhere and try to destroy your hypothesis. That's the way how should go.
Well, my original hypothesis was that even a small decrease in fidelity would be disastrous, and that was destroyed because that wasn't what I found.
And that was
It
actually
turned out that it was not the fidelity that mattered.
And that's very important as
Yeah,
well
yeah.
from my perspective because
Yeah.
from the model that I have, it is RNA-peptide world, which does not rely so
Right.
much on the fidelity of the replication.
That's right.
I wish to have similar models for just trying to model the RNA-peptide world as I have it in the paper. But if you wish, we can discuss later, of course. After,
Yeah, we can talk. I would like to talk about a collaboration. I think that would be great. Yeah.
So we can do that.
Yeah, and
So,
the peptide, the RNA peptide world, I think is a great idea. I mean, I don't know. You probably know the work of Sostak and his working on RNA replication using peptides, which at first it looked like it was going to go well, and then he had to retract it. But I think that that's still a very interesting idea that the peptide. It's somewhat different from what you're doing, but he's talking about the peptides getting rid of the self-annealing problem with RNA replication. But you're talking about stability. Sorry, I missed.
I would like to actually to start. So I would like to say lots of things, of course, to introduce what is all about. But first of all, I would like to kind of point out that there are lots of deceptions or fallacies in the origin of life, thinking or ideas and so on. The very first deception or fallacy was that life started and after that evolution kicks out. So that was in mind of many scientists, even today. Even today, a very famous scientist thinking somehow that life started, and after that, evolution kicks out and continue.
Right.
That's completely, absolute or wrong. It is fallacy. Fallacy.
Right.
So it is deception in origin of life. Why? Because evolution, Darwinian evolution, and life are the same thing. They are identical. You cannot start something and after that the other, simply because are the same thing. There are no such a thing. Life started and after that evolution started. Simply at the moment, once you have Darwinian evolution, then you can think for start of life.
Exactly.
That's everything. And this is the crucial point. And if you think about this crucial point is kind of sharp. It's kind of more sharpy point. It's not like usually when the first scientists in origin of life research, starting from Orgel, Jacques Zostak, Dimiter, and all of this population of scientists somehow back in their mind stays some sort of very gradual working on the first. You have evolution of chemistry, evolution, and after that you continue to have some polymers, and after that these polymers will start to do something, and eventually down the road you'll have cell, protocell, like let's say protocell. However, this is a very wrong concept, because this graduation of going, and they said, okay, so you don't have pretty much good boundary between what is life and what is chemistry. This last paper, it's exactly about that. It says very clearly, there is a very clear difference and very clear sharp boundary between chemistry and biology.
Right.
And this boundary is the moment when you have information dependent selection.
Right. I agree completely.
At that moment, once you have information dependent selection, that's it.
It's like the Big Bang.
It's like the Big Bang. Yeah. Something dramatic happened. It is sharp. I wouldn't say, so when it says sharp, it doesn't mean that it's life started Friday at 4 o'clock somewhere in the Haydn and something like that. Not sharp in this way. It is sharp as a phenomenon, it is sharp as interactions of specific polymers and specific moleculars. That's one thing. So this is the deception number one. And the people still didn't get it for some reason. And many hypotheses simply avoid even discussing this moment. And again, in the paper, it says very clearly, don't even say that you have hypothesis for origin of life if you don't come up with the idea how the Darwinian evolution came up. Otherwise, you don't have anything. You have just mumbo jumbo overall things. Yes, they might be good. They might be interesting. Yes, they might represent some sort of chemistry. Okay, that's good. We have to, by the way, I want to mention that we have to give our kind of gratitude to, to the chemistry scientists. They did a very good job. So we have for sure to thank to these guys. They did a lot of work on that. But even so, the moment is very crucial. The second deception, fallacy, in origin of life is the self-replication. Oh my goodness, self-replication. I mean, to some extent, self-replication is kind of entering the mind of any single scientist, every scientist that it's in the field, that it is kind of equalized. It's almost like if you're talking about origin of life, you must have self-replication. And that's a wrong, because self-replication, as you mentioned, as you said before, self-replication is something, is a phenomenon which is for the complex, for the entire complex, for a protocell.
Right.
Cells are self-replicating, yes, they are self-replicating, but you cannot have self-replication of single RNA. Right. It never happened even in the origin of life, but even by now. And because I'm coming from the field of DNA replication, so to me, replication is my field of science. And I can tell you by right now, there is not even one or single naturally, completely naturally to find self-replication in a RNA or in a DNA.
Right.
It's never happened. It's not in existence at all. And your model that you published it, actually demonstrate very clearly that it's not the case. Not the case. Of course, self-replication comes from the RNA world hypothesis, because the RNA world hypothesis, very shortly to say, it's you have a very short RNA, which is okay. You can have that. It's shown from our friends chemists that yes, indeed, you can have short RNAs. But in order this RNA, they start to interact, they must have some sort of ribozyme formation. Just for the auditorium, I will say the ribozyme is an enzyme which is composed entirely by RNA. There is no protein part. So the ribozyme formation, you have ribozyme formation, and after that there will be a moment which will have a ribozyme which will start to self-replicate. And once you have this ribozyme to start to self-replicate, now you have another problem, because from self-replicated ribozyme, you have to come to the point of RNA-peptide or RNA protein world, which we call that a polymer transition. This polymer transition must occur if you have only RNA entity. But in order to have that, you must have high fidelity replication.
Right.
So see what happened. RNA world by itself depends too much on replication fidelity. Right. That's why Jacques Zostek and Harvard, his team, they're doing everything in order to show that it's possible to have non-enzymatic template-directed polymerization, or non-enzymatic replication, to say another way, with high fidelity. And because they tried initially, actually back to Orgel, back in the 90s, has been done. And yes, indeed, such a replication was shown to be feasible, very well feasible. You can have replication which is non-enzymatic replication, a template-directed replication. However, this type of non-enzymatic replication, if it's on RNA, could occur only based on mostly by G and C nucleotides, guanine and cytosine nucleotides. When you add adenine or uracil, for example, the reaction stops. It blocks. Not to mention that such a non-enzymatic replication, it's prone to mistakes. So it's not with super high fidelity. And
yeah, I think their goal is like, at one paper I read, SOSTAC, they were looking to try to reach 70 or 80% fidelity, which is...
So what actually happened, they said, okay, it's not RNA, but probably something else,
Right.
like the RNA-like could do it.
Right.
And indeed, so you have approximately another 10 papers published with many students involved and many graduate students involved, defense of probably PhDs also involved, all of that. And they showed, yes, indeed, you can achieve such a higher fidelity with all of the 4 nucleotides, but not from exactly RNA. It's a RNA-like. And also these nucleotides have to have a specific conditions. They are not exactly the same nucleotides. I'm not going in details. It's not important for the discussion, these details. But to me, this is simply showing and that end of the origin of life. RNA world hypothesis simply, to me, from my perspective right now, it's just history. It's a nice try. Nice try. It was very reasonable try back in the 90s. But approximately 30 years of domination of RNA world. Now it's time to actually revisit it.
Can I break in for a second? Because I just want to mention that when I sent my paper in for review, I had a whole paragraph on RNA world. I figured, well, since that's so popular, I should include it. One of the reviewers said, take all that out because RNA world is no longer considered, the absolute consensus of what happened.
Yes.
And of course, he was right, and I did take it out. I still mention it, but yeah, there's so many problems with it, and they just keep getting worse and worse. So I think I agree completely with that, and it was a little too simplistic to begin with, I think.
And for example, for polymer transition, just for polymer transition, from, let me say, you already have self-replicated ribozyme to the moment when you have another system. Now we have translation. The polymer transitions, there is approximately 27 to 30 different hypotheses how just these polymer transitions should.
That's almost, I can't even begin to think about. I mean, I think you have some ideas about it, but I didn't get a chance to finish that paper, which talks about the origin of amino acylation. Yeah, the amino acylation.
Yeah.
But maybe you could.
Yeah. Can I have just 2 minutes and I'll be back, and you can talk with Tangelo maybe? So Tangelo, if you have some accumulated questions, we can you can present some questions to say. And also I would like to, of course, I will say a few things about RNA-peptide world, because so far we discussed about RNA world hypothesis. But I have to say a few things about RNA-peptide world and how this RNA-peptide world could be introduced actually in the model that say have his mathematical model that can be. Just give me 2 minutes. I'll be back.
Okay. Yes, what I think was very interesting, which Dimiter said, and which I fully agree with, is that RNA self-replication has never been demonstrated. Do you agree with that? Yeah.
Oh, absolutely. That's well known. What fools a lot of people, and whenever I say anything on social media, somebody always brings up the paper by Gerald Joyce, which says RNA self-replication in the title. But as Dimiter knows, that Joyce is a great scientist, but it's not actually template-directed self-replication, which is what we're talking about. What Joyce is saying in that paper title, when he mentions replication of RNA, he's actually talking about a different chemical process called ligation, where you have an RNA which gets bigger, and it's a wholly different process. It's not template-directed replication, which is how DNA works, for example. And the other thing that Dimiter said that's very important is that chemicals do not self-replicate. Cells do, but DNA does not self-replicate. RNA does not self-replicate. Proteins certainly don't self-replicate. DNA replicates, but with the help of a lot of other enzymes and proteins, and the same with RNA and proteins. People have brought up prions. No, that's not replication. That's a whole different process. So the only thing that replicates are living cells, not chemicals, not structures, nothing else. So, that I was glad he brought that up, because I forgot to mention that.
Yeah, and it's only DNA which replicates through the complex machinery, but since the entire cell has to duplicate, so these are all other very complex processes, like the organelles which expand, and then they duplicate in two parts, and so on. And the entire orchestration also to make from one cell membrane a second one, that is also all very complex, and that has to be explained. And I mean, even the first living cell, how did it do that? I mean, I cannot even imagine a way how that could occur.
Well, that's the problem. Now, what I like to hear, Dimiter has this very interesting hypothesis about the origin of the informational driven replication, genetic code, and the stabilization of RNA. So, and what he called, he's back. Okay. What he calls the RNA-peptide world, which is, that's very interesting, and it'd be interesting to hear more about that.
Okay, I leave you guys continue your conversations. Okay.
Okay, so I will share. Can I share my screen? Let's see. Oh, I didn't. Just 1 second. So I didn't pull out the paper.
Yeah, there is a share screen button down there, I see.
Yeah, just I need to open the right program. Okay. So here. Okay, so I'll share this screen. Okay, so can you see?
Yep, I remember this.
Okay, you see that? So, so here I would like to mention something very important. At least that was my case. When I started to think about what could happen and what cannot happen the main concept was not to tell the nature how to make life. That was my first idea. So I tried to take more like observational view, not to say what is that must happen or not happen. So for example, if I have to compare with RNA world hypothesis, they always, the scientists from RNA world hypothesis, they always used to say, okay, the nature must do that. It must prepare and make this and these enzymes, ribozymes. They must do that and that. But this is simply telling the nature what to do. I don't like this approach. I would like just to stay more like in observational situation. So to observe what could happen in such a conditions. And here with the lines, actually, in this particular slide, the lines presents RNA. And again, we have a very short RNA, not long RNA, because simply chemically you cannot come with very long RNA. Although it's possible for a very, very low percentage to have a little bit more and longer RNA, but not too much. So I'm talking for something like 5 to 10 nucleotides RNA. This is the straight lines here. And also the RNA, as everybody knows, can take a 3D shape. So it can make a shape because of Watson-Crick nucleotide interaction. So you have different structures and hybridization process. In addition, these circles or triangles or rhombs or diamonds, all of that represents the amino acids. And we know from the chemistry that the amino acids could aminoacylate RNA even without enzymes. I hope you agree with me in that.
Well, if they're activated, yeah, that's right.
Yeah, it is shown. Mm-hmm. And now it's quite possible. Also, it's very reasonable to see that some RNA is aminoacylated just naturally, chemically, right, without any enzymes or anything like that. Mm-hmm. But if you put this system in a temperature cycle, this is very similar to PCR, by the way.
Right.
Yeah, because in a very high temperature, you have denaturation of the RNA. So all of these hydrogen bonds are disrupted, melted. And in a low temperature, they start to hybridize back again. So you have a cycles of up and down the temperature. However, at some point, just because in a close proximity between the ends of the RNA, and this is due to hybridization reason, some peptide interaction could happen. Peptide bond. The peptide
usually, amino acid interaction giving a peptide.
Exactly. So this is happened. Why it's usually it is involved some kind of dry cycle in that case. So what you can expect in that situation? So up and down with the temperature cycles plus additional dry cycles, which is very reasonable to think about, you have formation of short peptides, which we call hybridization dependent peptides. This is not peptide just smashed each other because of some chemical interaction, but this is a hybridization dependent peptides, meaning formed because of the hybridization process of RNA.
Right.
So what you can expect from this particular hybridization dependent peptide, those peptides are not coded, that's for sure. Why? Because every RNA could pretty much have the same chance to interact with any amino acid. And also, the compositions of this peptide heavily will reflect the concentration of these amino acids. It is very reasonable and quite understandable. Most of the amino acids are alanine and glycine. And of course, the HDPs, these peptides, also will be mostly alanine and glycine. However, it depends on the concentration of amino acids. By chance, from time to time, there will be incorporation of another amino acids as well. And that's a logical to understand very well. Just due to this reason, some of the amino acids, like arginine, for example, they are prone to interact with RNA. And that I don't need even to prove that. Everybody knows that. It's shown experimentally hundreds of times. Mm-hmm. So, therefore, some with very low percentage, with very low probability, some of this hybridization peptide will interact with RNA. So you have an interaction with RNA and this peptide in a few cases, in some cases. So maybe 1 in 100,000. I didn't calculate that actually. You might help me to calculate exactly.
Yeah, we can do that.
In another case, we call it bridge peptide. The bridge peptide will contain two different than glycine and alanine amino acids, but simply because of the different electrocharged interactions. So they will attract, so we'll have attraction, electrostatic attraction between the amino acids. And in that case, in specific situation, some of those forces will help just to the close proximity to charge some of the RNA. So we call this particular peptides, which is even rare, even more rare than just interaction with RNA peptide, we call it bridge peptide. Because it's bridging, it's bridging RNA with the specific amino acid. So here I want to show something which is very important for our discussion right now. This is experimental paper which just came this year. And of course, we knew for similar papers before, but what this paper is saying experimentally, that interactions between peptides and RNA actually stabilize the complex dramatically. And it stabilizes it by more than 30 folds. It's here, it's written. So let's say between 20 and 30 fold stabilization process. And this stabilization is very important because that's what drives the next steps. So here I want to show, so for example, this slide, it shows how it's possible non-coded peptide to become coded. And that's very important. So you can imagine that you have bridge peptide assisted charged RNA here on the top left. Bridge peptide assisted charged RNA. And also, in addition, there is many random charged RNAs, all kinds of different charged RNA. Those will enter in a thermal cycle that I just showed before, like this one. No, not this one. So, not this one. This one. So they will be involved in those kinds of interactions, thermal cycles. So what will happen eventually down the road, they will be again by chance de novo synthesized HDP, hybridization dependent peptide, which will have a sequence like a bridge peptide. But those bridge peptides will be involved again into amino acylation of another set of RNA. Here it's very important. So let's see. This is the same picture as before, a slightly more detailed picture here. It's one to say that in position 1, you have a bridge peptide which interacts with RNA in position 2. In that case, I'm just repeating the same slide as before. So the previous things, whatever I said. You have electrostatic attractions between, for example, amino acids, which is negatively charged, and positively charged arginine. But that helps for amino acylation. See, in a position 2, you have amino acylation kind of slightly helped amino acylation. So this RNA will be amino acylated from arginine because of these bridge peptide interactions. Those complex, this amino acylated on a number 3 here, will be involved again in a thermal cycle and formation of hybridization-dependent peptides. But due to the reason that this particular complex between RNA and bridge peptide is more stable, it's stabilized, it will circulate longer. It circulates longer in the system. So it will be involved more frequently and more frequently into amino acylation process. And that, of course,
so I have a question that's, which is pretty important. So what you're saying is that bridge peptide would bind to one specific RNA, not yet a tRNA, but like a tRNA, because of its shape, because of its,
like
so in other words, because you wouldn't have, if that bridge peptide could go to any RNA, regardless of its structure. What's good about this slide is you show it fitting into the structure. So the idea would be that when it does hybridize, it's going to only hybridize to that structure of RNA. Is that correct?
So that's the moment which I need a little bit more explanation.
Okay.
First, there are two things. First of all, I have several evidence or clue, let's say clue or close to evidence that the primordial RNA is predominantly GC-rich.
Right.
The second thing, I have to mention that experimentally has been shown that arginine in complex with peptides interacts mostly with GC-rich RNA.
Right.
So by saying that specific RNA will be specific to the extent that it's mostly GC-rich.
Okay.
It will not be a specific super exact sequence, but still some GC rate will be important.
Okay.
Will favor these interactions.
Right.
And this is, again, I'm saying that it's shown that way. So if you think about this cycle from 1, 2, 3, 4, what will happen? You will have accumulation of more bridge peptide synthesis, because every single time you have arginine that will be involved with this particular RNA in formation of de novo bridge peptide. It's happened like a sequence, like a cycle which stabilizes the situation. So the question here is where the information comes from. What is the actual information? The information is this GC-rich RNA, which is more GC-rich, which will be linked to the arginine, free arginine in position 3. So you have link, although relatively weak, that's not the modern complexes. It's just relatively enriched, linked between arginine and GC-rich RNA. That's where the information is. And this system is more stable, as I showed you from the paper. It stabilized the system. So it became more stable and more stable. That's why you have also a structural component here. So the bridge peptide by itself will stabilize. Therefore, you have some kind of structural component which is linked to the informational component here. Therefore, this system is capable to information-dependent selection at that moment. Initially will be not super specific, but will favor to information-dependent selection.
Okay.
That's what I'm trying to say. Of course, next step, I can discuss next step, but it's more complicated. I can say that if you look the genetic code, for example, this is the genetic code distributed by GC richness. Based on everything that I just said, the very first RNA is mostly GC rich, and arginine interacts predominantly with GC rich RNA at the beginning. The code that will be assigned in the future, of course, in the future, will be GC rich code for arginine. And that's exactly what we see here. So if you just arrange the codons from GC rich to AU rich, you will make some sort of chronological idea for the assignment of the different codon formations.
Right.
Of course, this is a little bit longer discussion, so I wish to go in details for that. But right now, we don't have that much time. I just want to show the basic steps, logical basic steps to formation of LUCA. Of course, this is what must happen without knowing the details. We don't know anything here. So the very first step where you have non-coded HDPs to the coded HDPs, the Darwinian evolution emerged.
Right.
As I mentioned, this is a relatively sharp moment. Darwinian evolution started from then and continue after. The very short peptides actually can do a few things initially, but not much. Predominantly, at that moment, this short peptide can do mostly first to specify the bridge peptide and to show and to demonstrate specificity, relative specificity, which amino acid will interact with which RNA. And also, this is very important about replication. And we have to mention that because it's not just important, but it's part of your formula, your model about the replication. Every single polymerase which is existing today, polymerase, actually have as a catalytic center. The catalytic center is just 2 or 3 magnesium ions. So you can imagine how simple is that. If you have positioned the magnesium ions in a specific distance from each other, that will facilitate the replication process. So it is extremely reasonable, a short peptide, to hold magnesium ions, and that to facilitate the initial, very raw replication process. But the peptide, this is now peptide. So how we can think about the replication before that moment? The replication before that moment was just non-enzymatic polymerization process template directed polymerization. It was the same way as it was shown in the 90s from Orgel, for example. They showed that it's possible, but mostly in GC-rich RNA. It is possible some of the components, some of the small motifs of RNA to be maintained just because there is such a non-enzymatic replication. But this is the moment until you have a short peptide that can hold 2 magnesium ions. Or some bivalent ions. It's not absolutely necessary to be magnesium in details, but
Right.
the idea is, you see the idea what it is.
Mhm.
So at the moment, once you hold these 2 or 3 magnesium ions, now that facilitates the process of replication. So that will be the second function of the motif of the peptides at that moment. But this is the very first moment. During that time, all the other functions will depend mostly on RNA. This is why we call this RNA-peptide world, because RNA is still the master here. This is the moment where the ribozymes are selected to be ribozymes, because the peptides are too short. They cannot have too many functions. They are not. It's hard to imagine that those short peptides can involved in, for example, maturation of tRNA. So, in fact, exactly RNA is involved in maturation of tRNA, as we know today from the ribozymes. That's why you have selection of ribozymes. At the same time, there is evolution of ribosome and the entire transcription apparatus, which we don't have right now time to go in details. But the ribosome by itself is a ribozyme, which has been selected exactly in that period of time. And once you have folded long peptides and folded proteins, now you have formation of catalytic centers, and at that moment, as you know very well, the functions and the ability for another functions are almost unlimited. So I'm stopped sharing right now. Of course, there are many details, and for example, my second paper, which is very important about the formation of genetic code, formation of the very early ribozyme, all the steps that follows logically, I'm saying logically follows about that. We can discuss it with hours. So right now, obviously, we cannot do that.
I do have one question about the model, though, going back to the charging of the amino acid onto the RNA from the
bridge peptide.
The bridge peptide. That requires energy to get that amino acid onto that.
Yes.
To the end of the RNA. So what is the source of the energy in this model? Is that some kind of ATPase or?
No, there is no ATPase.
That wouldn't work, right? So how does that happen?
The source is actually very easy to figure it out, because during the prebiotic chemistry, most of these precursors, like nucleotides, amino acids, all of that actually are synthesized not in a clean way. They are synthesized activated. They was already activated from the heat source, from the source. So you have activated, as a chemist, you know very well that the curve is like going up, so up, going up and down. So you have activated energy.
The activation energy, right?
Yeah. So this activation energy was pre-activated from the synthesis of this nucleotide.
So that's with a
Or
phosphate or some other high energy?
there are different variants.
Okay.
We don't know exactly,
No,
but it
could be.
has
It could
been
be.
shown. Yeah, it has been shown that activated, they don't simply does not come in the clean form like purified from sigma Aldrich
Right.
nucleotide. So,
Right.
and that is just a rough idea of the model that
So
was.
okay. So it's not a big problem, is what you're saying, because
I don't think it's a problem at all.
Okay.
Although every time when you're talking with, I mean, not talking, but when you read for metabolism first, origin of life researchers, they try to make a very big deal of that.
Right.
I don't think it's big deal at all.
Okay.
Otangelo, any questions that we can discuss?
Yes, to be honest, I've been more involved in the side chat to answer some questions, so I've not followed the conversation. So I'm sorry, I don't have any questions right now.
To get questions from the chat?
Yeah, has someone from the chat, side chat, any questions in regards of the conversation?
So if someone, so that's what I learned from the teachers around me. If there's no question, if there's no questions after your presentation, that means two things. Either everything is absolutely clear and nobody has questions,
or nothing was
clear. Nothing is clear and there's no questions at all.
Yeah,
that's right.
I mean, the guys in the side chat, they have talked about evolution and other topics, and I mean, I could have restrained them and say, hey, it's off topic, listen only to what Dimiter and Sai are talking, and then ask them something, but I didn't want to be so restrictive and leave them to talk whatever they wanted. So speed here.
So I'll be, give me 1 minute. I have to watch. I'm watching something. That's why I have to go. Just and I'll be back in 1 minute. So
okay.
Ask the questions. Yeah.
Okay. Let's see if someone is asking something so we can clear it up.
Why did God make creationists? Well, actually, this discussion, it's very technical. It's true, and the reason is because, at least from my point of view, and I think Dimiter feels the same way, we're both approaching the same question from different angles, but it actually has a lot to do with evolution, because as I always say, abiogenesis, you didn't get evolution to create life. Okay, evolution comes once you have life.
That is something which I think you should address that to Dimiter, because I strongly disagree with him, because he puts evolution into the abiogenesis scheme. It says that evolution already happened before life started and DNA replication.
Not quite. It may look that way, but that's not actually what he's saying. What he's saying is that some of the steps in the abiogenesis to Luca involve evolution, and I also don't agree with that. I'm on your side on that. Okay. I think Luca doesn't come from evolution, but that's a theological, philosophical view. However, he also agrees that first line, that very sharp border where you don't have Darwinian evolution yet, that before that, we don't know how we got to that line. I mean, he's back, so we could ask him. That might be a good thing for you to ask, or I could ask.
Yes, Dimiter, as you know, we had in previous interactions where I pointed out that I disagree with you, that evolution was a driving force before life started and DNA replication. And Sai corrected me and said that your position is that just some things eventually emerged by evolutionary forces. Maybe you can clear that up and elucidate what your position is exactly.
Yes. Again, two things I forgot, and it is related to the question as well. So first, I forgot to mention where the RNA-peptide world hypothesis, as this one, enter into the Garte's model, because in your model you have a probability for stability,
Right.
and also you have which is called PS, and also you have in your model FR, which is the fidelity of the replication. So how the peptide world actually comes and could be plugged in your model. In fact, if you see the model, the interactions between peptide and RNA increase dramatically PS.
Mm-hmm. Right.
This is the moment where you increase dramatically PS, so the probability for stability. For the fidelity of replication, it's a little bit more complicated. What I mean, it's not very clear here, because in fact you have only, let me say, between 2 and 4 nucleotide system, which is important. And this, let me say, 4 nucleotides out of 5, 6 nucleotides are only important for the process to go. But those are predominantly GC. And if you apply that to just non-enzymatic polymerization, template-directed polymerization, you will see that simply the combinatory effect will result in a relatively, I'm saying relatively high fidelity. Not higher, but relatively high, because simply you have just a few nucleotides, and those few nucleotides are mostly GC rich.
Oh my God.
Anyway, so that will result relatively high fidelity of the replication.
Okay.
So from the question that was about the where, so how to start the Darwinian evolution will start. Actually, here is the moment. Everything before information-dependent selection to start, everything that is formed as a complex will have the equal probability to form and disappear, to dissolve every time. Therefore, there is no upgrade. There is no upgrade. So you have interactions of all kinds of molecules, all kinds of interactions with different species from everything, everywhere. That might form a relatively stable complex, just by reason of polymers, for example. Mm-hmm. But those complexes will form and dissolve with the time with the same probability, and every time will be formed with the same probability in the same conditions. But, and here's the big moment. Once you have information-dependent stabilization, every time you keep memory from the previous stage, you keep memory from the previous situation. Right. That's why you are able to upgrade the system. The system can become slightly better next time.
So what your model is proposing is a form of what you call upgrade, which is like evolution, because you get this stabilization which carries over to the next stage. And so it's reinforcing the stabilized products but it's not actually Darwinian evolution in the sense of how we think of it in terms of natural selection. And
No, it's not.
it's not. It's a different it's a chemical evolutionary process.
So this is a question about terminology. In fact,
Well, yeah.
what is important here is not the terminology, but the
Yeah.
important is that you have bridge peptide, which, because it interacts with predominantly GC-rich RNA, next time you stabilize predominantly GC-rich RNA, next time will be the same, and that increase. So you have a moment of memory. Although this moment of memory, it's in a very kind of simple complex. It's a super simple complex, just a few nucleotides and a few amino acids. That's it. It's nothing more than that. It's a very simple. That's why I call this. That's why I call this organism Terra Darwinia. The name of this organism is Terra Darwinia, meaning the very first system which is capable of Darwinian evolution. So if you call
but I'm not sure. But you say that the term doesn't matter, but I think it. I don't think you could call it Darwinian evolution. It just has a different mechanism. It's stabilization rather than selection. I mean, there's some selection involved, but it's
stabilization.
It's a different
information-dependent stabilization.
That's true. Yeah, that's true.
That's it. So if you call it, if you call it Darwinian evolution, if you don't call it Darwinian evolution, I'm calling it Darwinian evolution because it's kind of how the term it spreads among the scientists.
Okay.
That's why it is so. So Lina have a question. I heard something. Hi, James.
Yeah, I was going to say, I'd love to hear what James has to say about all this.
Hi, yes, I've only met Cy and interactions on chat, and Dimiter I only saw 2 days ago on Speed of Sound and Gravity's channel, the interview that RNA was in. So I'm sorry I haven't had a chance to read your paper yet, but enjoyed your presentation. I'm a biochemist, so my questions are more biochemistry related. But I guess to stay on topic of what you guys were just talking about, and the whether it's Darwinian selection or not, it sounds like the selection process is you're favoring the more stable molecules, but in doing so, does that not also restrict the variability that you would need to get the complex types of polypeptides? Like if you're favoring glycine and alanine, which are both non-polar, aren't you going to end up with a bunch of polypeptides that have mostly hydrophobic nature? And if arginine is used as one of the stabilizing factors, presumably because that guanidino group that is usually positively charged at less than was it pH of 13, I think, wouldn't that also precipitate with some of the anions that are present in the primordial soup, like chloride ions and phosphate? And most of the reactions you've shown, of course, are using the known biotic structures, but in a realistic, prebiotic world, you're probably looking at 10,000 to 1 of abiotic, different molecules that have nothing to do with life, so they won't help, and more frequently they will actually hurt because you showed the amino acyl group binding to the hydroxyl group on your proto tRNAs, but just as easily that that carboxyl group on the amino acid could form an ester with that hydroxyl, which would cut down the at least in half. And then you have to think about the tens of thousands of other molecules that contain amino and carboxyl and other alcohol reactive groups that are going to get there first. And so when you're trying to build your fledgling polypeptide, a specific sequence is needed because, as you said, information, the selective pressure doesn't happen until you've got some kind of sequence or information. But as soon as it gets 3, 4, 5 long, in goes not only the wrong amino acid, but the wrong molecule. You could have some abiotic molecule attached to that acceptor stem on the tRNA and put it on the growing chain, and then you no longer have a functional protein, let alone something that would reach the length that would get the complex three-dimensional folds to perform function. So it seems like you're selecting for very homogeneous types of polypeptides where it favors just certain amino acids. And arginine is another problem too, because I was looking at a paper, review paper that came out 2 years ago by Katadai and Mariyuma. I can't pronounce their names. It's called Origin of Building Blocks of Life, a review, and they have a nice some tables in there that summarize what sorts of amino acids out of the 20 proteinaceous amino acids have been found in extraterrestrial samples, carbonaceous chondrites, as well as what sort of origin of life primordial soup type experiments have yielded the various nucleotides and amino acids. And arginine, as far as I can tell on their list, there was only one paper in 1976 where they were able to produce arginine, and it hasn't, according to that paper, been found in any of the meteorites or interstellar media. So it's questionable how much arginine would be available in the primordial soup. So sorry, I had a lot of questions in my mind. I got a few more, but I'll let you sort through those first.
So a few things I addressed in my second paper, actually, the question about the low level of arginine. And without any doubt, we don't have any doubt about that all of the models, simulation models of prebiotic synthesis shows almost none to some arginine. Maybe not all, of course, but arginine is very reasonable to be there. If you see, for example, the one of the latest review paper of Susilov and Sutherland in 2020, they reviewed very well that arginine is quite expected to be there. That's one thing. But anyway, even that it's quite expected to be there, it will be very low as a concentration. And that's the trick. This is the moment which is I also described in the second paper relatively well. Usually, all of the hypotheses for origin of genetic code, especially if you look at the papers from Higgs, he promoted the idea that you have abundance criteria, meaning the first, he actually ordered the amino acids based on abundance. And he said the first 10 of the most abundant amino acids, and arginine is not there, of course. If first 10 amino acids, the most abundant, are involved, that's how the life originated with only these 10 amino acids because was most abundant. Now, I'm arguing in the paper, in the second paper, that the abundant criteria is important, but not everything. Meaning, because you have active selection of the peptide which contains arginine, and this peptide actually it's predisposed the situation when recruit another arginine. It's kind of selection, information-based selection. Now it is quite possible, it's logical also, to have a recruitment of lower abundant amino acids into the system, just because it makes more stable. So it's not necessary to think about that just because some amino acids are more abundant than the other are not important. Of course, we don't know. I can say we don't know. This is a moment of experimental, we need to provide a lot of experiment. About the this is favored to specific, so some kind to uniform the peptide that will be selected, as you mentioned. Yes, to some extent will be, but nothing is perfect. And don't I have to mention that there is a now the next function will come somehow, meaning. So, for example, in the second paper, which I think it's one of the most important papers, actually, from all of the 3 papers, because it describes very clear that the first incorporated amino acids, which is assigned as a codon, which is alanine, glycine, arginine, and proline, those amino acids are with GC-rich codons. Therefore, those are supposed to be the first one. And in a complex on a very different variant, they might influence very well the further stabilization of the complex. I should not, I didn't mention that at some moment, more advanced form will appear, simply because there will be a separation of the functions. So this is more like developed in the first paper, 2018 paper, I developed that. You can expect that more advanced form will, the different RNA, for example, will start to take slightly different functions in this system. And because of this, now you have selection of more and more variety of different amino acids. Now, as an honest scientist, I have to say, we don't know. We don't know all of the chemistry, all of the sequences, all of that. This is still hypothesis. However, even so, this hypothesis is prone to experiment. This is not hypothesis that cannot be experimented. Can be experimented, and I'm sure there will be lots of surprises there. I mean, no doubt.
Yeah, thank you. I'm still a little unclear on what sort of selective process occurs when you're creating these short sequences of polypeptides. Let's say one just happens to fold into a protein with a function, but what if that function is like a decarboxylase or a deaminase or something that could be detrimental to the neighboring polypeptide chains? And then how is that molecule and the information or the process of continuously generating more of that molecule so that it somehow gets filtered out from the rest of the permeations of possible sequences of amino acids? What we're talking about before the cell. So what kind of advantage do these random, semi-random? You're saying they're selected based on GC content of the tRNA complexes that help bring everything together, but there's still, it still sounds like the selection is going to be based more on the abundance of the amino acids that can stick onto these proto-tRNAs, and 2, the GC content of those tRNAs, and not so much what does that protein actually do to help bring us closer to the first cell.
So you're talking for too far away. So initially, I'm thinking only for this very simple complex. First, I have to say one thing. It's important. We assume that this system will occur in some sort of compartmentalization. So you expect to have some chambers, some either lipid layers or some sort of those kinds of things. So you will have relatively close system. And that's natural. I mean, that's understandable as well. Second, it's from that moment, we have to think evolutionary. from that moment. Simply, if something helps and stabilizes the system and makes it more adaptive to the system, it will survive. I'm absolutely sure all kinds of proteins, at that moment you don't have proteins. You have just a very short peptides. And even this RNA that I showed, although we can call it proto-RNA, at that particular initial moment you have vice versa. Either RNA will serve as a template or serve as a proto-RNA, or all of that. So the same RNA can serve either way. At that moment, everything it's not settled down completely. A little bit later, a step later, they will be slightly more kind of going toward RNA, which is more like dedicated to tRNA, and another one that will be more dedicated to serve as a template RNA, as a ribosomal, for example, RNA. It's simply the selection thinking from that moment, only the process which was selected. So as you said, the filtering process, I even don't dare to think in details at that moment, because from that moment, the system became too complicated to predict exactly what it is. I wish to do that, but I cannot, because unless I don't do experiment and I don't show how the things are going and what kind of sequences are selected, I simply don't know. Yes, I mean, it could be so from my perspective, if peptides happen to be, let me say, some kind of, let me say, peptidase, degrades the peptides. Well, if such a thing occurred, it will not emerge. It will not stabilize. It will not stabilize. With stabilize only things that will survive. That's why you have many other species, many other interactions there. All of that will happen for sure, but eventually we are talking for something that after all emerged. It is complicated at that moment. And I say, you cannot predict it, I cannot predict it, and I don't think anyone can predict it. What kind of sequences could emerge at that situation. What I can say, this is entirely feasible for experiment system. So it could be experimented.
Do you think that the types of ribozymes or polypeptides with enzymatic function, are they more likely to sort of evolve into functions with bond breaking capacity or bond forming capacity? Or, is it 50-50, or is it, we typically think it's easier to break something than build something. But I was trying to think, in a primordial soup situation, would it be more likely that we would have endonucleases and exonucleases and proteases rather than, ligases and polymerases? And
well,
that would be a problem. All you need is one enzyme to act, especially if it hasn't developed the ability to regulate. There's no inhibitor present to just totally wreak havoc inside of a protocell, some kind of compartment, or even if it was let out. I did some research with ricin toxin using just the lectin part to deliver insulin to the GI tract, to the gut-associated lymphoid tissue to induce oral tolerance. But I remember reading about the A chain, which we didn't include that gene because it forms an enzyme that detaches from the B chain. It was just a disulfide bond that holds it. And so the B chain, the lectin, helps attach it to pretty much any cell of the human body. And once inside the cell, it releases the A chain. That will just cut off one adenine base off of the ribosomal RNA, and it will deactivate like 1,000 ribosomes per minute. And that's what kills the cell so fast. It just shuts down translation. So if something like that were to emerge in a primordial soup, and all it did is recognize one particular sequence and start cutting it. It seems like it's almost inescapable that that is going to win in a sort of Darwinian survival, where it's the only thing standing after it destroys every other fledgling growing RNA or growing DNA or growing polypeptide.
So, I mean, this is completely legitimate questions and worries. However, I'm judging by, for example, the ribozyme, which is the RNA-P ribozyme, which is involved in the processing of tRNA, for example, and that's a ribozyme. It is absolutely kind of logical to think about that those ribozyme, although in a highly primitive form, could occur at that moment. And this is actually cutting ribozyme. It is cut to produce the tRNA. So, and also the second, the ribozyme is the PTC center of the large subunit of the ribosome. And this PTC center could be just very simple, like a ring style, simple. And because there is already hybridized processes, anything that facilitates the peptide bond, like this ring style, the proto-PTC center, will dramatically improve the system, will dramatically improve. Those kinds of things could be expected. I know they could be peptidize occur, could be all kinds of, but see, this is a very short peptide as well. So at that moment, that first moment, which is RNA-peptide world, you have just a few peptides, not more, which is functional peptides, which is preserving the system. And all of the rest is the work of RNA. That's why it's resemble RNA world, but it's not RNA world, definitely. Because RNA world is entirely entity of RNA and there's no peptides, it's appeared later. And you must have self-replication and those kinds of things, which I completely disagree with that model.
Yeah, I agree with you, especially, with the high rate of hydrolysis of RNA. I remember my colleagues down a couple lab benches down from when I was doing my graduate research, they did a lot of RNA work and they had to be so fastidious with their lab where they just, they used mirror surfaces so they could make sure no one touched it because of all the RNases that just come off of the body and trying to do these RT-PCR. Yeah, and even without the, even when they used RNase inhibitors, just leaving it in the solution, aqueous solution in the fridge. I mean, we know with these coronavirus vaccines, the mRNA vaccines, they have to be kept at below minus 70. Once they're solubilized in water and stored even at refrigeration temperatures, they only last 1 to 4 weeks, and that's after Pfizer and Moderna, spent quite a bit of time figuring out how they could put a biologically safe coating. They use lipid nanoparticles that I think come from pig, but and even
glycol, maybe they contain ethylene glycol, maybe.
Yeah, that helps keep ice crystals from forming and pulling the water of hydration off of there as well. But I guess the hydrolysis problem is still an issue, even when they're coated in these sort of protocells.
Yeah.
The RNA world, I think one of the biggest problems is you're always going to have a certain amount of water for these chemical reactions to happen. And yeah, you can have dry cycles, but eventually it's got to get back into being in an aqueous environment to participate, to fold in the proper three-dimensional structure. If it was catalyzed on clay, it's got to come off of there so it can have that correct 3D fold and perform its function. But with proteins, I don't know what the smallest size protein is that still has function. Almost all of them are going to be like peptides, like nanopeptides, oxytocin, that require cooperation with very large receptors. So I don't know how you would get a short functioning peptide that has very complex activity. I don't know. Do you know of any short peptides that?
Let me say just 1 second. this is the situation of simply evolutionary thinking. You definitely, I mean, short peptide cannot have any other function than whatever its a few amino acids contains. Just amino acids, if it contains hydrophobic, for example, amino acids, will be prone to go and interact with lipids, for example. Or if it's contained positively charged amino acid will interact with negatively charged surface or RNA. And that's quite understandable. Nothing else you can expect from peptide. The peptide is peptide. It's a small and function just, the function is very limited. That's why at that moment you have selection of ribozymes to do something.
So in your model, the peptides are there just to act as stabilizing agents,
Exactly.
to interact with the membrane and to interact with the ribozymes until they can get to the length. But again, how does the ribozyme even get to the length it needs to? What do you think it takes? Like 50 bases long before it can fold into a secondary or tertiary structure?
The ribozymes do not need to be too long in order to do whatever they do initially.
I think the hammerhead ribozyme is one of the shortest ones, but it has multiple RNA strands as part of it. But again, because it's so fragile, and in your model, you talk about having a warm cycle to melt apart these GC-rich regions to denature it. And of course, it takes more energy when you have a higher percentage of G's and C's. That's what the extreme thermophiles, their genomes have a much higher GC concentration because of the 3 hydrogen bonds instead of the 2. Right. And then it has to cool down to an ideal annealing temperature so you don't get non-specific binding of other molecules that can hydrogen bond. So it will have to be at just the right annealing temperature. And then there's also a drying cycle to concentrate everything and reduce some of the hydrolysis. I mean, this geologically would take some time, wouldn't it, to find a region where the pool can raise in temperature, come back down to an ideal temperature, and then dry out to facilitate the peptide bond formation. And but eventually get back into aqueous solution where it's got that uphill battle with Le Chatelier's principle with hydrolysis, trying to destroy everything that was so preciously made in the past cycles.
First of all, you have to think about. Let me switch on my light. But this is the world without much oxygen, free oxygen around. So that helps a little bit. The second, ribozymes that will be selected initially, it doesn't mean that should be the full developed ribozymes as we know it today. I'm talking only for the natural ribozymes, which we know exist as a natural ribozymes, not the ribozymes that imaginary ribozymes that was synthesized in the laboratory. Now, don't forget that at that moment, you already have Darwinian evolution. Therefore, everything that contributes to the complex will be stabilized. So everything starts to be explainable. Although we don't know the detail, we don't know lots of details, it's still, you are already into Darwinian evolution. So
I'm sorry that I lost my audio. I couldn't hear anything for the last 15 minutes. So you may have already covered this. Sorry about that. I had to get out and get back in.
Yeah.
But I'm sorry if I asked something that you've already talked about. But the problem I have with you saying that this is Darwinian evolution is back to what we talked about at the very beginning, which is that, with Darwinian evolution, you're talking about a continual change, which is progressive, presumably, which is brought about by selective advantage and by replication. In other words, I don't see how you get Darwinian evolution unless you have both of those characteristics. Now, stabilizing is great because that will allow those RNA molecules to persevere and, as you said, go through another cycle. But where is this occurring? Is this occurring in a cell? Is it occurring in random space? If it's occurring in a cell, at some point that cell's going to have to divide and allow the same stabilizing process to occur in both daughter cells. Or am I missing something, or would you discuss this?
Before start to answer, I'm just thinking about your question. Let's switch my light.
Okay.
And, and I'll be back.
Sorry, I don't know what time it is over there. Probably getting tired. Of course, we could go on to this conversation forever. So anytime anyone needs to go, we can always.
Yeah, no, I'm sorry. I'm sorry I missed that. When I don't know, it was quite a while before I got my audio back, so I missed.
So is it better? Not too much, but
it's all right. We can.
I'm still visible. So.
Sorry, I don't know what time it is where you are, Dimiter. It looks like you've been talking for 2 hours, so I don't mean to wear you out.
Yeah, he's around 5 o'clock. 5 o'clock, but it's still getting darker outside. Yeah. And I'm in a construction process at the moment, so not everything is set up. So
for me, you guys can go on. I have no problem. So whenever you want to finish, that's
we can finish it soon.
Any constraint. Yeah, I'm not giving any constraint. Whenever you guys want to finish, that's fine.
Yeah, I want to answer to Sai whatever he said. So I don't know weekend terminology used as a terminology exactly Darwinian evolution. So I used this terminology because it was kind of accepted as a terminology in the field. For example, if you take the NASA's definition of life, it says that life is a self-sustaining chemical system that is capable of Darwinian evolution. Oh, and also I've seen a lot of scientists in the field to say that this moment of Darwinian evolution, even in that initial state, we can call it Darwinian evolution. However, to me, that doesn't matter exactly. What is matter is that it's information-dependent stabilization.
Okay.
So if you look the third paper, the last paper, I provided the definition for Darwinian evolution.
Right.
And this is a dynamically stable system, which is where the information determines its own supporting structure.
Okay, so that's the other question I wanted to ask, which again you may have already discussed while I wasn't able to hear anything, and that is what you laid out. I thought was very interesting, and I agree is the beginning of an informational structure because it's GC rich. Okay, but as we know, that's the very beginning of the informational structure, and when you compare that to the genetic code, where you have really very sophisticated informational structures, not just GC rich, it's specific, anticodons. Yeah. There's a big gap in my mind between GC rich versus AU rich and actually having, GGG as an anticodon on the RNA. So do you go that far, or your model is just starting with the beginning there on the GC rich, and you don't go how you get to the next step?
Yeah. First of all, from Terra Darwinia, which is the very first system that is capable of Darwinian evolution or information-dependent selection.
Right.
So from Terra Darwinia to Luca, this is a huge, enormous space of time.
Right.
There's so many things happened during that time, like a selection of tRNA, mRNA, ribosomal RNA, evolution. So you have a process of co-evolution of tRNA, mRNA, and rRNA. And they are co-evolved in order to fit and to provide better fidelity of the translational system. And those should happen, although the primordial ribosome could be just a few loops for the large subunits, which I explained predominantly presented the PTC center, and a few loops for the small subunits, which is only today you can project that in a helix 44 in the small subunits. This is the very first position where you have interactions with the codon-anticodon interaction. However, at the very beginning, these interactions are very primordial and very weak, meaning they're not with high fidelity, but still providing advantage. And after that, you have, so in the second paper, again, I explained in more detail. So if you go in the second paper where I provided for the evolution of the genetic code, I dividing the LUCA formation in two major steps, two major periods, which is pre-LUCA 1 and pre-LUCA 2. In pre-LUCA 1, which is the actual real RNA peptide world, you almost after, when it's finished this period of time, you already have kind of shaped the ribosome.
Okay.
Although it's not completely, it's shaped the ribosome. And then, of course, you continue to put more and more loops and more just to advantage. If you think about, for example, in the ribosome, EFTU is the factor which is involved in the recognitions of the third codon base or the first anticodon base. So the same thing. Mm-hmm. And there is a so-called clutch mechanism, which is the codon and anticodon, they fit each other, and only when it's clutched well, so you have, when they fit perfectly well, it clutched, and the EFTU then it's release the RNA into the PTC center. However, if you don't have this mechanism, because this EFTU is a protein, it's a very advanced protein.
Right.
If you don't have this mechanism, what you'll have simply better recognition of the first two nucleotides, not the third one. And if everybody knows that the third one is the wobble.
Right.
So that's how it works. So it initially works mostly on the first two nucleotides, and the third one came after. So during the pre-LUCA 2. I don't know details. I mean,
there's a huge gap between GC rich and even the first two nucleotides in the codon. Okay, that itself is, I mean, what you've got to propose, I mean, I can imagine a scenario where you have shapes of these proto-tRNAs which bind to specific binding peptides, and they happen to have the correct anticodon on them. So that is the formation of the original genetic code. The problem is there's so much room for error. And that's where my model about replication fidelity comes in. That the system can only handle a certain amount of error and continue to go forward, whether you call it evolution or whatever. So I'm a little worried about that. That's a very large missing gap in my view, is how you get, an actual, even if it's a 2-base anticodon, how do you get to any anticodon with all this difficulty in getting accuracy?
Especially when you look at what needs to happen today, where you've got 20 different aminoacyl tRNA synthetases. One
that's not even right.
to recognize the right tRNA and the right amino acid, and then there's a dozen other
where did they
enzymes
come
that are
from?
involved in preparing that tRNA as well, which require energy. And energetic molecules just have short half-lives, so you have to be able to replenish them.
So just to finish our discussion, because it's getting too long, I probably will finish with one thing. At the moment, there is two hypotheses for origin of genetic code. Two types of hypotheses for origin of genetic code. One type of origin of genetic code postulate or hypothesize that with every single nucleotide from the codon, so 1, 2, 3, brings the next set of amino acids. So meaning if you have just, let me say, one nucleotide codon, you have only 4 amino acids. If you have 2, you have next set of amino acids. If you have 3, you have all the set of amino acids. So this is the first type of hypothesis for the origin of genetic code. However, the second type of hypothesis of the genetic code is that you pretty much have all of the 3 codon anticodon already developed on a level of few, only 3 or 4 assigned amino acids.
Right.
So, and that gives expansion of the next assignments. Because of the fidelity, it starts to increase. But the very first level, the very first amino acids are just set up for the for all of the 3 meaningful, although third is wobble, so it's not too much meaningful, but let me say mostly the 2 nucleotides and the third wobble, it's set up only on the first assigned amino acids, and I can name it. This is arginine, alanine, glycine, and proline. After that, it's coming the serine and threonine. So, and guys, that's the science. I will say that's a terra incognita. We don't know a lot. I mean, this is something between Terra, Darwinia, and Luca. It's an empty space. We don't know what happened there. I mean, just because we already provided some mechanisms of explanation, it could be explained. It needs a lot of research and simulations to go there.
Yeah.
Just as you were talking, I thought of another dilemma. The bend in the anticodon has to be just the right angle so that only 3 of those bases are exposed. If it was a little too loose, we might have a quadruplet codon, or too sharp and where there'd be just 2 of them. And so the fact that it's always 3 on every one of these tRNAs, you showed a diversity of sort of tRNAs that wouldn't necessarily have that sharp bend that exposes just 3 of the bases there, so that they can interact with the messenger RNA. Yeah. There's so much in between those gaps.
Yeah, don't forget the fully developed tRNA, the fully developed small ribosome, small cell units, the large cell units. It came just in the second stage of pre-Luca 2. When you already have a very high setup the system, meaning it's already under evolution a long time. Also about the cell or not cell, as I said, this system cannot be in a free, in an open ocean. Right. I mean, we know that. Those system is expected to be developed or to happen in some sort of compartmentalization events.
Right.
Microchambers, lipids layers, or sediments, or something which
between 2 crystals.
Yeah. So, for example, the basalt, after the volcanic basalt, you know very well if it's in Hawaiian today, you can see it directly. The basalt is full of micropores, a huge amount of micropores, and that could be easy chambers for many things. And who knows what? I mean, I don't know exactly what kind of microchambers could occur there, but definitely should be there.
Well, there has to be a transport mechanism, or they get entombed, but that's a whole other discussion.
James, I would prefer to stop at the moment if there's no some questions, but I would like to keep communication because obviously you are a biochemist. So if you don't mind, we can exchange emails and continue. And with you say, Garte, I hope we can continue discussion. I can send you email also.
I have your email. Yeah, we're all on the same email thread. I don't know what it's called, but
so I would like to thank you to Otangelo that he brought us scientists here. It's a food for thinking. Although nothing, the science does not work with ready solutions. It's a process.
I really appreciate your time. I've been interested in abiogenesis only a short period of time, maybe last 6 months. So I'm still trying to play catch up. So it was nice to get an update on your model, and I'm sure we'll get to exchange some more. But yeah, it's fascinating. I love learning new things. I guess I get bored with just doing the same thing all the time, which is probably why I've created 15 different science courses in the last 10 years that have kept me busy, and I'm finally having time to go back and catch up on things that I'm interested in, like abiogenesis. So I appreciate your time and explanations.
Yeah.
I have put in the side chat and in the private chat a link to Ron Stadler's assessment to abiogenesis. And I think it is a great lecture. I would encourage all you guys to have a look at it. He has also a book, which is Stairway to Life. And he points out several problems in regards of abiogenesis and things that I didn't know, like homochirality in DNA. There are many different forms of possible chiral forms, not only left and right-handed of ribose. And I think it was a very entertaining and interesting conversation. So thank you very much to everyone. Thank you also to those which have been assisting the conversation. And I would appreciate if we can keep it going and have other conversations like this. I think it is very educational as well. So all sides are being heard in a respectful manner without over talking and so forth. So I really enjoyed it, and I thank everyone for this conversation.
Thank you so much. Bye, guys.
Thank you, guys.
So I will end this conversation in this stream here, and see you guys next time. Thank you very much.
Okay.
Bye-bye.
Thanks, Angelo. Bye-bye.